odpoledne. Dovolte, abych vás všechny přivítal na dnešním přednáškovém odpoledni nebo hodince a půl, která je věnována problematice difuzního středočarového gliomu. Já si moc vážím toho, že jste přijali naše pozvání a jste tu dneska s námi. Jsem rád, že to pozvání, jak vidím, přijali vědci, klinici, ale i taky veřejnost a že vás tohleto téma zajímá. Jsem hrozně rád, že se nám podařilo sem přitáhnout kolegy z Francie, z Institutu Gustave Roussy, který se této problematice věnuje opravdu velmi extenzivně v Evropě a patří mezi ty nejlepší týmy, které ve světě jsou, a bych si dovolil už teďka pokračovat v angličtině. So, hello once again, welcome in this lectures concerning diffuse midline glioma. I'm very happy that I can invite colleagues from France, from Belgium, Institute Gustave Roussy, a special team which dedicates to the problematic of pediatric brain tumors. And this lecture series is called Diffuse Midline Glioma from Bench to Clinic. I'm very happy that I can invite Dr. Samuel Abu, Dr. David Castel, engineer Anne-Florence Blandin, and magistra Denisou Olejníková, která s nimi strávila více než 1 rok a ještě stále s nimi spolupracuje na této problematice. I will give the possibility to the first speaker, with Samuel Abu. Thank you. Samuel Abu is clinician and also a scientist who dedicates to the problematics of pediatric brain tumors, and he's going to take first of all.
Thank you, Martin. It's more than you. Sam, that's my name. I'm a pediatric oncologist, and I focus on brain tumors and precision medicine and new drug development. So we try to, because we are a team working together in the labs, but also with patients. So we try to do something broad to show you from the clinic, from the lab to the clinic, but I will start with a little bit of clinic because maybe not all of you know about this disease. And so that's why we called it from bench to bedside. This is my conflict of interest. So this is a slide that Jack Real, who is leading the IPT program and Gustave Roussy, gave me. It's just showing a really old book from 1945 about how they describe the IPGs, and this is a really, really old slide of how it was at that point. What is important is to understand that those diseases were for a while described as a clinical disease, clinical and radiological when you got radiology, but this is really old. And those are called midline gliomas or diffuse midline gliomas. And I think the important thing, and what is really different from most of the other brain tumors, is the infiltrated part of this disease. Because this disease, it's not like a big spore or something that you can remove. It goes in the normal brain, which gives a big problem to the surgeon, because the surgeon, they cannot remove the brain itself. So they cannot operate it. They can do some surgery, some biopsy, but the surgery cannot be possible, and it's a very specific disease. For a while, you see so like a little bit forward in time, 1993, we used to say the diagnosis, and when I started med studies, the diagnosis was clinical and radiological. Just saying we have a clinical history, short one, because you have low-grade gliomas that can be at the same place, and with imaging characteristics. And everybody used to say, you don't need to do a biopsy in the pons. So, in the pons, so that means between the brain and the rest of the body, is too dangerous because the patient cannot survive it. So nobody did anything for it for a while, and we knew that radiotherapy worked, but that's it. But it worked just for a while, and then unfortunately, the main patient, the disease progressed. The MRI gave us a lot of information about this disease, and I can show you many different images. And this is where you start to think, but if there are many different images, you told us it was just one image and a specific clinic characterization. So is this the same disease? And this is where it gets complicated. So you can see here, that you can see a cystic part of the disease in the pons. So the pons is here, the brain is here, and you have the spine going here. So the pons is really important, and you can see that the disease is here with a bit cystic, a big bubble in it. But I told you about the infiltration part, and here you can see the white is the infiltration of the disease, but you can see the white going here and going here. So this is important because you don't see exactly where is the disease. The disease is infiltrating, and you know that the MRI is not perfect to see the cells. So it's more than what you see. And we can also see metastasis in this disease. Sometimes it's midline glioma, so it can be the pons, but it can be the thalamic, and thalamic is here, and we can have a lot of necrosis, and sometimes we can have major infiltration and the disease spreads everywhere, like from close to close palm. And we know that the best treatment for now, and it's like not huge, a very good treatment, but the best treatment for now is radiotherapy. You can see before radiotherapy and response to radiotherapy. However, the problem is the disease doesn't stay, and David will talk a little bit about that. It's not you do radiotherapy and then it's cleared. You do radiotherapy, it's cleared for a while, and it depends a lot on a lot of different things, but then eventually it comes back. And so we were at the clinic, then the radiology, and then a lot of things came, and you see it's recent, really recent. I mean, I guess that everybody was born here, not from 1945, but here everybody was born. So you see new definitions with subgroups, with histone mutations, with other kind of mutation, with other kind of DIPGs, etc., etc. I don't go into all details here, but you can see that things have changed a lot. And still here, 2023, like 2 years ago, we still discover things, and we have so many things to discover. So for a while, I said it before, the biopsy was largely debated. But you can see that without a biopsy, you cannot say anything. So between 1993 to 2013, we don't put any dates because basically nobody was doing any biopsy, nobody was doing anything, so we don't know anything about the disease. And when people started to do biopsy, so the people who started to do biopsy, I have to say the name, is Stéphanie Puget in the Necker Hospital, when at that point, people from, Jacques Grille used to tell her, you need to try, and she was, okay, let's try, and we'll do it. Stéphanie Puget is an amazing neurosurgeon from Necker Hospital, and why biopsy is that important? And so people used to tell her, you're crazy to do that. But she managed to do it and to do it safely. I will show you how. But this is, I think, the main reason. We need to learn a lot about the disease, but you look at these two diseases. You can see a pons infiltration, and you see the infiltration here. You can see a pons infiltration, and you see infiltration here. And I will tell you one thing. One of these two tumors is not high-grade glioma. One of these two tumors is low-grade glioma, with a completely different treatment, with a completely different outcome. This is rare, but it happens. And it's enough to have one patient that has a different thing that to justify a biopsy. So the biopsy, how to do it, basically it's this system that allows the surgeon to put exactly where they want you, based on the imaging. So they calculate with the imaging where to go, and they calculate the exact distance, and they go exactly where it is. It's called stereotactic biopsy. Neurosurgeon, I really train, some neurosurgeon I really train for that. In Necker, they do it every month, so they're really, really trained. It's important to be trained to know where to pass and where not to go. And with that, we have this mutation, and this is something that is very important in this disease, because it then gives you all the information that you need to understand the disease. This disease, so this is just, I won't go into details, but you can see the histone mutation. So the histone, what are the histones? It's something that helps to fold DNA. And those mutations, basically when you see euchromatin, I know that there are geneticists here, so I'm trying not to say stupid things. So you have the euchromatin and the heterochromatin, and the histone mutation changes the portion of one and the other, and especially it creates more, you can see that you can have the DNA that is completely open and you can read it, and the DNA that is completely like a whole pellet and you cannot read it. And basically the histone mutation, when it happens, it's H3K27, now it's called 28 or whatever. You can see that it prevents the methylation and it changes this structure of DNA. And when we discovered this, we discovered that it has impact on the aggressivity of the aggressiveness of the disease, and you can see that some histone, the histone 3.1, are different from the histone 3.3. This curve shows you when the patient relapses and when the disease is stronger than the patient. So you can see the difference between those two. Even if for both of them, eventually the disease will come back, you can see that there is a difference in terms of time. And this is important because it helps us to think about why there is a difference and can we use this as a treatment. I don't want to go into details, but that's one of the things that we are working on as a new drug development, targeting specific instability of the genome, and we can target it with specific drugs. And you can see here, just showing this quickly because then my colleague will present that part. Just showing that what we can do is, with those instability that we will working on, we can add drugs and test the drugs. And so I will let David continue. I've been told that I'm too fast, so.
Thanks a lot. Very happy to be here. I'm Taurus. Sorry. And thanks a lot for the kind invitation to show a bit what we do in Gustave Roussy. So I will step back a bit and just, so I'm basically heading the lab with Jacques and working with all these people here. And I just wanted to explain a bit how we do research in our team in Gustave Roussy. So we basically have research that's really centered on DMG. And also other tumors, as I will tell. And we try to have an integrated research program on this tumor, and we try to attack it as a whole, let's say. And we have project on oncogenesis and the tumor complexity, the vulnerabilities, meaning how to target these tumors, trying to identify new treatments, and also understand resistance to these treatments. And also working on this tumor in the brain, in the organism as a whole, because this tumor is not a ball in the brain, like Sam just said, it's infiltrating the brain, and we need basically to understand how it's interacting with the brain. So, sorry, in the strategy we have in the lab is due to the strong interaction that we have between the lab and the clinical department. And what we do is basically, we often start from the patient with patient biopsies or liquid biopsies, as you will hear. So surgical biopsies or liquid biopsies, as you will hear today. And we do a lot of analysis on this tumor. Genomic analysis, multi-omics, because a lot of layers. We try to get the maximum of information from rare cases. These are rare tumors, too frequent, obviously, but still rare compared to other cancer. And we try to correlate this data to clinical data, to therapeutic response, imaging data, to get all the specificities of these tumors, and trying to identify new subtypes, as I will tell you a bit today. And what we do also in the lab is basically trying to model, to develop avatars of these tumors, from patient material with cells, with organoids, with mice models, because we need these mice models to understand also the penetration of the drug into the brain and also the interaction with the normal brain. And with this, we can identify new targets, do some drug evaluation, and we can inform basically clinical trial. And that's really a cycle that we have in discussion that we have with our clinical clinician colleagues that are really fruitful in this context. So going back on DMG, basically we've been working from the first biopsy by Stéphane Puget towards trying to decompose the heterogeneity of this disease. And we could identify discrepancies that Samuel mentioned. Also tumors that don't have the histone mutation but have something else that is basically similar to the histone mutations. And I will showcase two examples of what things that we did in the lab to try to understand better this disease. And I will talk a bit about irradiation. At that time, the starting observation was based on these discrepancies between a 3.1 and a 3.3 mutated tumors. You can see that the light green here was more aggressive than the dark green with a 3.1 mutation, but also we could see that the response to radiotherapy, radiation of the tumor, was much better in the case of this A3.1 mutated tumor. And we wanted to understand why these A3.3 were poorly responding to treatment. And we started, so this was a clinical observation, and then we went to the lab and tried to do some in vitro work based on the cells that we got from the neurosurgeon and growing in the lab. And basically what we did, it's quite simple. Either we let the cells grow and they increase their confidence, they fill up a dish in the lab, or we do irradiate the cells with a dose of 2 gray or a higher dose of 4 gray, and we measure the delay in the speed at which they grow in the dish. And from this we can infer with mathematics, whatever, the surviving fraction post-irradiation to see the sensitivity of the cells. And we did this for basically an H3.1 tumor, an H3.3 tumor. It was behaving well. The H3.1 was more sensitive to irradiation, and then we extended this to many cells. And what was interesting is that we could see differences in terms of response to radiotherapy, and this was correlating basically with the time to relapse in the patient. So between the in vitro here and the patient behavior from which the cells were taken, we could see a correlation. The more the sensitive the cells, the longer the survival without relapse. And so we went further and we looked at H3.1 and H3.3, and it was behaving well. H3.1 were more sensitive, but for one. And some few cells with H3.3 were basically sensitive and not resistant, so there was something wrong in that story. And we went further, and that's basically the same thing. The higher the bar, the more resistant to the treatment the cells from these different patients were, and we looked basically at the mutation in the DNA of this patient, and what we could spot basically that it was not dependent on the type of mutation in the patient, on the histone A3.3 or A3.1, but it was linked basically to mutation in TP53, another gene that is known in cancer, and that is more often found basically in this A3.3 subgroup. And you can see here that all TP53 mutated tumor were basically more radioresistant in vitro. And having this in hand, we went back to clinical data, and we analyzed a cohort of 73 patients with basically clinical, radiological, and molecular study of the tumor to see if this was true. And basically what we could see is that indeed the TP53 wild type that are often A3.1 as well were more sensitive, a lot of improvement after radiotherapy, and same on the MRI, we could see a strong response to radiotherapy, and it was not the case at all for the TP53 mutated in both clinical and radiological response. And basically, what we could see very different here was not significant comparing it to online. And well, these are statistics to show that basically TP53 mutations are really independent markers of this sensitivity to the treatment. So basically, we could show that TP53 mutations were really predicting if the patient would or not respond to radiotherapy, and this is basically a problem. I mean, that's a prognostic factor that is not used in clinical trials. In BioMini2 trial, there is a randomization that is dependent on TP53 mutation, meaning that we try to give the two treatments that are compared to the patient that have mutation or that don't have mutation, to compare these separately for the effect of the tumor and not to have confounding effect. So I will skip. And I will just continue with another story, still working on the heterogeneity of this disease, but this time it was more on the invasion and the extension of this disease in the brain. So basically, as Samuel said, these DIPG patients with pontine tumors are diagnosed. You can see with the blue arrow that they have a tumor in the pontine region. But they get irradiated, some respond a bit less, that's what I just showed before. But also, what happened is that some of the patients will progress or relapse locally at the same location of the primary tumor. Some will relapse around the primary tumor, what we call local regional spread. And some will have tumor all over the brain with craniosspinal or let's say intracranial metastasis. And you have 1/3 of the patient with local, a bit less than a half with local regional, and 1/4 that are metastatic in the cohort that we have. And we were wondering why does this happen, and can we explain and can we predict this? So first we looked at what it was doing on the patient survival, because a lot of people were telling us, well, DIPG is a diffuse disease, it goes all over the brain, and there's no heterogeneity in that context. And if you compare the local with the local regional with the metastatic in red in terms of survival, if it goes down fast, it means that the patients, unfortunately, are dying faster. You can see that it's a pretty bad prognostic factor in this context. And we could also see that, not considering metastasis, but just the speed at which the disease will spread on the MRI, and that's basically a radiological invasion that's in centimeters per day. It was basically inversely correlating with the survival of the patient, meaning that the tumor that spread very fast would kill faster. So there's definitely a need in this context to understand and to try to counteract invasion in the image. So we took a cohort, I'm sorry for the text, it was messed up by the Mac PC. So we took a cohort of patients with local, that's in gray here, local regional in black, and metastatic evolution, and we did the sequencing of the tumor to try to find DNA modification that could explain why the aggressiveness is different between the patients. And basically what we could see that all the mutations that we identified, H3.3 and H3.1 mutation, TP53 mutation, PPM1D, ATRX, whatever, all these mutations, all these genes could be found in all categories of patients, and we could not understand what was the driver of invasion. So basically we went back to the lab, and again, comparing primary tumors with models, avatars, and we, from these patient-derived cells, we did some spheres, glioma spheres, that we did embed in, let's say, that's a matrix, artificial matrix that can a bit recapitulate the brain microenvironment and constrain the cells in their spread. And what we did is what is called a sphere invasion assay. We would take movie of this, and we would record basically the speed at which the cell would invade in that matrix. And from the speed that we record with the microscope, here that's this one, and the blue one is that one, we can compute an invasion score. And we did this for 22 patient-derived models, and you can see that we got quite a big heterogeneity in terms of invasion scores. And when we looked basically at which patient was where, because again, these are cells coming from patient. We could compare the speed of invasion of the patient to the evolution in this patient. And basically what we observed is that the faster cells would belong to metastatic patients. So here you have the score, the higher the score, the higher score basically correspond to metastatic patients. And this is something that we try to reproduce now in biomedia to try to predict the appearance of metastasis in patients. But we wanted to know why, basically, and what was the biology behind. So basically we took these cells again, embed them in a matrix that resembles the brain, and grew them for 3 weeks before taking what we call the RNA, just to have a snapshot of what the cells express in terms of protein, an RNA profile. And we had 3 local, 10 local regional, and 9 metastatic patients. And I will cut these analyze this short, but what we wanted to identify is basically genes that would increase their expression with the invasion score, and basically this is what you see here for BMP7, bone morphogenetic protein 7, that is low, that's the expression here, in low invasion scores, that's the blue samples, and that increase basically with the invasion. And what we did next is basically we have tools to try to remove one protein from these cells. And basically when we did this, we compared the control with the targeted BMP7 cells, and basically in several models we could decrease this invasion, meaning that it was not only correlating, but if you reduce this protein, basically it slows down the extension of the cells. Looking again at our expression data, we looked at the correlations between BMP7 expression and some expression profile in the cell, some signatures that are known in cancer, basically to provide invasiveness. And interestingly, EMT, epithelial mesenchymal transition, is usually a process that you see involved in metastasis. And in our hands, it was kind of funny because EMT was low. It was low in the very invasive and high in the non-invasive cells. So, and when we looked at these cells, indeed, the low invasive were looking very mesenchymal, cells that spread and that they get upper, and to migrate, they would grasp basically the extracellular matrix, the protein that are in their environment. And in the case of high invasive cells, you have very low grasping, but they have a contraction of what we call the actins that you can see here, especially at the rear, and it's propelling the cells. So instead of a crawling cell, you have a propelling cells. I'm making the analogy because we are in this museum. And so basically we went a bit further, and we could indeed see that these cells had a lot what we call focal adhesion, where they grasp the environment to crawl, whereas there was no grasping in these cells that were highly contractile. But what was very interesting is that we can really shift from this mesenchymal low invasive phenotype to the very highly invasive amoeboid-like phenotype just by putting BMP7 protein or by removing BMP7 protein, meaning that we can really switch between two modalities, a low invasive mesenchymal or high invasive amoeboid-like phenotype. Sorry, it's stuck now. And now we are wondering basically if the low invasive are stuck in the bones because they are trapped by the stiffness of the brain where the tumor is, whereas the very invasive basically don't care about the stiffness. They just crawl around and they don't need to grasp, and they don't get stuck, and they go very far away in the brain, and that's the working hypothesis that we have presently in the lab. And obviously, what we were interested in is trying to target BMP7, and I will cut this long story short, but what we could see is that there's a receptor for BMP7, BMP receptors in a range, and basically when we did put BMP receptor inhibitors, we could decrease the invasion speed of these cells, and what we identified is that the signaling of this inside the cells goes through proteins that are called MEK, ERK, and ROC, because each time you would target MEK, ERK, and ROC, we would decrease basically the invasion speed, meaning that there is in the armamentarium of oncologists some protein that could decrease this expansion of the disease, for example, using MAPK inhibitors such as trametinib in this case. So these were just to showcase two examples of what we do. We work also in the lab on other diffuse gliomas and ependymomas that share similarities in terms of oncogenesis, resistance to treatment, or invasion processes, basically, and we work also on some predisposition syndrome. And just to very quickly for the audience to know what the type of project that we have presently running, we're really working on tumor heterogeneity and the different subtypes of tumor that we have, and trying to identify also the heterogeneity within one tumor with the different cell states. We try to model early oncogenesis, the very early event that leads to a derail from the normal development in the brain of the kid. We think it's even starting in utero. And we also study a lot of therapeutic responses from the biomedi trial, given that we have responses to treatment. And we have also the analysis of the tumor to try to find explanations for the response or resistance to treatment. Also, we work on trying to overcome radioresistance, that's the follow-up of what I showed. And we try to identify new vulnerabilities with genetic screens mostly, and trying to do drug development from these newly identified vulnerabilities. And last but not least, we are still working on invasion, obviously, a lot to do still. And we also work on trying to put immunotherapy in this tumor, even though it's going to be tough, because immunotherapy is not doing anything at this stage on this tumor, except for some few results of CAR T cells. So the pathway still will be closed. Stopping by thanking, obviously, and showing you all the people from the team. We are a team of 20, 25, depending on when, with great scientists, great clinicians, postdocs, PhD students, and technical people, and thanking collaborators and funding for this job. Thanks a lot.
Hi everyone. Thank you for giving us the opportunity to show you our data here. That first slide is like a transition slide. I just want to, so with some, we are developing this liquid biopsy program. Liquid biopsy as think about fragments of tumor that are released into biological fluids, and we are developing that with some area. This is the people that are. So this is the people that are I'm just not going to look at the screen. Okay, good. So here are the people that are so far involved in this program, and the Linda is here today. She's gonna show you some of her data. So I think at this point of the presentation, you guys did understood that those tumors are very challenging, and there is several challenges that remain in clinic these days. Just to cite a few of them, the biopsy samples that do not reflect tumor heterogeneity because they are performed at one single location. They perform in dangerous locations, sometimes they are also not accessible for surgery. Another limited another problem in the pediatric brain tumor context is that so far we have limited tools to detect minimal residual disease and undetect only relapse. And last but not least, that's something that I'm sure you're aware of. It's very hard for the drug to penetrate, to go inside the brain because of this physical, this really physical barrier with cells that are very tight and close to each other, and they prevent the drug to get into the brain tumor. So we do need to find a better way to study tumor evolution, better way to detect cancer recurrence, and new strategy to improve drug delivery. So how do we overcome these challenges? There is several options, but one of them is called transcranial focused ultrasound treatment. So the ultrasound treatment is nothing new. There is several medical applications that have been expanded since the 90s. Back then they were using the heat or the thermal effect of ultrasound to remove specific parts of the brain in the context of movement disorder or Parkinson's disease. And much later, the medical community started to think about ultrasound treatment as a way to temporarily open this blood-brain barrier that I was just telling you about, with the idea that that can increase drug delivery, that can sensitize tumor to radiotherapy, that it can potentially treat epilepsy, and also increase the access to liquid biopsy. So these fragments that are shed from the tumor into biological fluids. So our goal within this liquid biopsy program in David's team, it's to detect these tumor brain derived circulating biomarkers using mouse model and patient sample. So the first step was to do a feasibility study. How can we use available tools to open the blood-brain barrier in mice? And we are using this preclinical mouse device commercialized by this company called based in Paris. We are collaborating with them. We are focusing, when I say brain-derived circulating biomarkers, there's plenty of biomarkers you can think of. But we needed to decide, we needed to choose, and we need to do the proof of concept. So somewhere we decided to go with what we know, and that's DNA and cells, escaping from the primary tumor. And the second aim is to evaluate how we can combine this ultrasound treatment with existing drug and treatment, and would that make it better? So that's what we are going to talk about today. Why all of this is important? Because if we are successful, we could elucidate the mechanism by which this ultrasound would help release biomarkers. It's not very well described in the field so far. We could determine the optimal time point to collect this circulating biomarker from the tumor. We could, as I mentioned before, evaluate minimal residual disease. So far, the tools, the imaging tools that are available are not resolutive enough to do that. So can we use circulating tumor DNA, and that's Denisa's work, you're going to see that in a moment. Can we use this biomarker to evaluate and predict tumor recurrence? I also told you that we decided to extract DNA and CTC as two types of biomarkers. We can profile the CTC, we can try to see what they are, or what they express, and how they change over time. We can try to make 3D models. That's something that's well developed in David's team, but not with CTC yet. But we can, benefit from their expertise to make it happen with a CTC-derived in vitro model. We can try to see if we can integrate these two types of biomarkers. So again, DNA and cells, and do they correlate? It's not always. It seems to be obvious, but it's not always true. Not in every tumor type. But as a general idea, I think people in the field would agree that circulating tumor DNA is coming from apoptotic or dead cells, but that's not always true, and we don't know if it will be true in the context of pediatric brain tumors. And then, last but not least, again, maybe uncover a new promising, therapeutic combination, ultrasound plus drug that are known to not get into the brain so far. So we developed this workflow to collect, again, DNA and CTC, but from one single tube, and that's somewhat important because you're talking about pediatric brain tumors, so you're somewhat limited by the volume of blood you can get from this patient by opposition to adult. And then CSF and blood, it stands for two types of biological fluid, CSF for cerebrospinal fluid, so brain fluid and blood. And from when we get this sample from the clinic, then we proceed to some, like centrifugation, spin step in the lab to extract the DNA. First part, extracting the DNA and like to perform ctDNA circulating tumor DNA study. That's the Denisou's project. And then from the same tube, the remaining blood layer, we will by a few steps that I will explain later, but we will collect, isolate cancer cells and try to profile them to see what they can tell us. And with that, I'm gonna let the stage to Lisa.
Good afternoon, everyone. To be honest, I never talked to this big audience before, so I feel sorry for my nervosity. Okay, so let's go. Today I want to briefly introduce you a project which I'm working for over the past year. It's focusing on circulation tumor DNA. In this project, we are analyze two types of samples, liquid biopsy samples, the plasma samples and the cerebrospinal fluid. In these samples, even in the healthy body, in healthy people, we are able to analyze to detect small fragments of DNA, which called cell-free DNA. And in the patient with the tumor, bude to jednodušší, když budu mluvit v češtině pro vás.
Určitě, já to připravil.
Nevadí vám to. Dobře, takže v tomto projektu analyzujeme dva typy vzorků, vzorky plazmy a vzorky mozkovýšního moku. Je důležité podotknout, že vlastně i v zdravém těle v těchto tělních tekutinách cirkulují krátké fragmenty volné cirkulující DNA, vlastně, které se tam dostávají díky apoptoze a dalších buněčných pochodů buňky. A v případě vlastně pacientů s nádory se tam dostávají tyto krátké fragmenty také z právě těchto nádorů a ty se my snažíme detekovat. Vlastně díky tomu, že je to velmi malá koncentrace těchto krátkých fragmentů a také protože máme velké pozadí té nenádorové cirkulující cell-free DNA, je to strašně těžké najednou mluvit v češtině, já se omlouvám. Potřebujeme velmi vysoce citlivou metodu na tu detekci těchto na detekci. Proto používáme vlastně digital PCR, což je vlastně ten přístroj, který vidíte na obrazovce. Tento je konkrétně od Scala Technologies. Všechno se děje vlastně v tom čipu, který vidíte uprostřed. A vlastně hlavní princip, proč je vlastně tato metoda tak citlivá, je díky tomu, že rozdělíme tu reakci na tisíce mikroreakcí, mikrokapiček, a každá kapička se amplifikuje samostatně, individuálně, a díky tomu vlastně zvýšíme tu citlivost. Na konci té analýzy vlastně ty kapičky, které obsahují tu nádorovou DNA, kterou hledáme, tak vyzáří fluorescenci. A vlastně výsledek analýzy vidíte v tom grafu nalevo, kde máme vlastně graf, kde vlastně každá ta tečka na tom grafu znázorňuje jednu tu kapičku v tom předchozím obrázku. Takže ty kapičky, kde byla nádorová DNA, ty vidíme v zelené barvě. Ty vyzářily zelenou fluorescenční barvičku, takže vlastně jsou ukázány jako zelené. Dole vlastně v tom šedém klastru, v tom ráčku vidíme negativní DNA a vlastně v tom modrém oblačku tak tam vidíme normální DNA, to je vlastně ta, co cirkuluje každému z nás v krevním objemu. Dobře. A v rámci tohoto projektu jsme vlastně vyvinuli dvě eseje, 2 testy, které vlastně používáme. Díky tomu, že používáme vlastně technologii odstevy, tak můžeme multiplexovat, tedy použít více, nebo detekovat více genů v jedné reakci. Máme vlastně napnuty 2 testy, 2 eseje. Jedna je vlastně pro klinickou studii nazvanou Sonoky. To je vlastně, ta se zabývá vlastně otevíráním mozku hematoencefalitické bariéry pomocí ultrazvuku. A druhá esej, o které budu dneska mluvit, tak je vlastně určená pro děti pacienty a vlastně obsahuje 5 genů. Z toho jeden z nich je právě ten referenční gen a potom vlastně jsme schopni detekovat histon 3.3 nebo histon 3.1, ACVR nebo TT53. Na tomto slajdu bych vám ráda ukázala, jak vlastně výsledek optimalizace a validace této eseje, že vlastně ráda bych vám první vysvětlila, co tam máte vidět. Tak vlastně, jak jsem říkala, jsme schopni multiplexovat všechny geny do jedné reakce, tak proto tam máte vlastně grafy pro všech 5 genů. Vlastně v prvním, tam, kde je sample 1, v prvním vzorku, tak tam máme vlastně pozitivní kontrolu pro histon mutaci a TP53. V vzorku 2 je vlastně pozitivní kontrola pro histon 1 a ACVR 1. A jak můžete vidět, tak po všech validacích a optimalizacích je tato metoda velmi citlivá, senzitivní, dokonce je specifická. Okay. Pardon, musíte mě omluvit dneska. Takže to je výsledek optimalizace této eseje. A tuto esej jsme použili na vyšetření několika pacientských vzorků. V levém grafu vidíte výsledky pro mozkovýšní mok a v pravém pro plazmu od pacientských vzorků. A ještě bych vám chtěla vysvětlit, že vlastně v modrém sloupečku vidíte vlastně ten referenční gen, což znamená, že to je ta normální cirkulující DNA. A v barevných, v zelené a v červeném sloupečku vidíte vlastně zastoupení té cirkulující nádorové DNA. A jak můžete vidět, tak vlastně v mozkovnějším moku jsme schopni detekovat tuto cirkulující nádorovou DNA. A v plazmě jsme prozatím nebyli moc úspěšní, máme vlastně jenom jeden záchyt a to si můžeme vysvětlit vlastně dvoumi způsoby. Jeden z nich je právě přítomnost hematoencefalické bariéry. Druhá možnost je to, že nemáme dostatečně citlivou metodu. Mám tady ještě jeden příklad, který jsem bych vám chtěla ukázat. A tohle jsou výsledky od jednoho pacienta. Vlastně výsledky plazmy a mozkovnějšího moku, kdy, jak vidíte, v plazmě jsme detekovali spoustu normální DNA v modrém oblačku. A v mozkovnějším moku jsme, přestože jsme detekovali spoustu normální DNA, tak jsme nebyli schopni detekovat histomutaci, když to v mozkovnějším moku, kde vidíte, že koncentrace celkově byla mnohem nižší, tak jsme detekovali histomutaci v 27%. Což je vlastně jináčí příklad toho, co jsem říkala předtím. A proto jsme se rozhodli, že zkusíme zvýšit citlivost naší eseje, a to tím, že vlastně před digital PCR ještě přidáme nějaké cykly preamplifikace, tedy že zesílíme ten signál toho vzorku ještě předtím, než to vložíme vlastně na ten digital PCR. A jak vidíte, tak po všech validacích, optimalizacích, tak jsme docela úspěšní. A v prvním sloupečku vidíte, že jsme byli schopni detekovat přibližně 100 pikogramů nádorové DNA. A po preamplifikaci jsme schopni jít dolů až na 1 pikogram nádorové DNA. To stejné pro druhý histon. A tím bych chtěla tuhle část uzavřít, že vlastně digital PCR se zdá jako dobrá metoda pro detekci těchto mutací a také, že preamplifikace nám pomohla vlastně zesílit tento signál. A v dalších krocích bychom rádi zjistili, nebo chtěli bychom vyšetřit vzorky po otevírání mozkovnější hematoencefalické bariéry a jestli jsme schopni detekovat vlastně histomutaci i v plazmě. A také bych ráda chtěla poděkovat obou stranám, jak nadaci Nina, tak kolegům z Institutu Gustave Roussy za jejich podporu během tohoto roku. A je to super zkušenost.
I've never heard about this project in Czech. It's nice. So going back to
Talk French.
yeah, now I can do it in French. So going back to these cartoons that will show you preliminary results of circulating tumor DNA. Now let's see what we have so far on the cancer cells level. I just wanted to emphasize that this tumor cell enrichment process, that's a step that we needed to add in our workflow, because if you consider blood sample, those circulating tumor cells are very, very rare. You need to think about billions of red blood cells, millions of white blood cells, and then depending on the disease and the context, but between, I don't know, 10 and 100 circulating cancer cells. So you need to find ways to increase that ratio cancer cells to rest of the cells that you're not interested in. And one way is to do tumor enrichment. So we try different methods. There is plenty of methods on the market, and we selected the one that we are the most happy with. And another step is to label, to stain with fluorescent marker these cells to detect them easily, to find a way to detect them more easily. And the last step in E here is something we haven't done yet. We are not there yet, but the ultimate goal is to do some genomic profiling of these cells to see what they are, why do they escape the tumor mass and not the others, and try to understand the biology of this tumor a little bit better. So when I was talking about the staining part, so if we zoom in on that step here, the first thing you need to know is what are they? Like what do they have at their cell surface? What do they express? And what can we target? So from there, you just like read literature, talk to your clinical colleagues, and try to understand which target would be the best one depending on the disease. So here is an example for the GLPGs. And so we tested several markers by fluorescence. Like like each marker here, each target, it's one of these dots. And then the question is, does the cell express that marker? Is the dot present here or not? And depending on disease, you don't have the same panel of these red dots. But for DLPG, we did select those targets. They are involved in multiple biological processes, but they all have in common, like all these targets that we tested, they all have in common that they have been described to be one way or another involved in brain tumor malignancy, in invasive behaviors and characteristics. And the double positive and negative selection, that's again, going back here, you have a lot of background cells, a lot of unwanted cells that you don't want to profile, that you don't want to isolate and focus on. So this positive or negative selection, that's an easy way. You pick two colors, you keep it very simple. All the cells you don't want are going to be stained in green, and the cells that you want, the cancer cells, are going to be red. And if you do that, this is an example of two patient samples, for example. And we did find, so we collected cerebrospinal fluid samples, so not the blood that I've been talking about, that's another biological fluid that we can collect from this patient. And those two examples are from this liquid circulating outside the brain. And this patient had the histone mutation that you've heard of with David Castel earlier. And we did identify, a bunch of what we think are circulating tumor cells here and here, because they highly express, like they were very red when it comes to this panel of cell surface marker. So then the next step now to fully validate that they are indeed cancer cells, and what kind of cancer cells is that, is to do the genomic profiling. That's on the way. The last part of this liquid biopsy program is the mouse work and this blood-brain barrier opening using the ultrasound technology. So here is how it looks like, this preclinical device commercialized by this company, Terrasonic, based in Paris. So the mouse is here, and the idea that comes with this ultrasound treatment is to inject into, that's true for patient, and that's also true for the animal preclinical device. You inject what we call micro bubbles, so they are literally very, very, very small bubbles, and they get into the mouse circulation. They are here, and then you start this ultrasound stimulation, and the ultrasound will make these bubbles oscillate, vibrate, and by doing so, they are gonna untight. Here is this blood-brain barrier, very tight junction I'm talking about. You see that nothing can go through. And when you use this technology, it's somewhat, like untight, and the shear forces like untight these cells, and could potentially let drug through, and also circulating biomarker being released into the circulation. So here it's just because it's a very cool technique, so it's getting into the details a little bit for you guys. But so we are, using a catheter that we place into the tail vein of the mice, and the mice are under anesthesia here, and they are like stuck in the convention system, so they are sleeping and they cannot move. And then we are using an ultrasound gel, and the transducer is here, and that will like transmit to to the mouse skull the ultrasound stimulation. Then you inject this microbubble I was talking about here, and you perform this ultrasound stimulation, and then you inject fluorescent marker, whatever you choose, to measure if this blood-brain barrier is open or not. And here is another little picture because the mouse starts to turn blue, so I don't think you guys have seen blue mouse previously. So that's another, there is multiple, clinical marker that you could use, but that one is this one is easy to use for us, and that helps us to validate that the catheter is well placed into the tail vein, and if it is well placed, then the mouse turns blue. If the mouse turns blue, that means that when you inject the microbubble, you're good. That means that the ultrasound stimulation is going to work, and that means that the green marker that you inject will be able to show you if your treatment was efficient or not. So that's just multiple way to triple check that we are doing things the right way. Here I'm not going to get into the specifics, but this is how the program, the computer looks like. And that's just to show you that you can do this ultrasound treatment at one single spot if you want to, but you can also decide to scan some area of the brain. So that's what's happening here. Like this ultrasound stimulation goes like this, and you do 40 times this repetition on the mouse skull, and that's about 2 minutes. And after that, the blood-brain barrier is open for some time. So the next question is, how long does this blood-brain barrier stay open in mice? This slide is quickly, I just have 2 or 3 more slides, and then I think we are somewhat in time. So here, just getting into the specific, maybe a little bit on why did we pick this green marker here. Most people do use MRIs in mice too, to validate that the blood-brain barrier is open. That could have been one option, but then something much faster and much cheaper is to use this green marker and to do fluorescent measurement of that green marker in mice. It's also a good marker because it's a small molecule, fluorescent molecule, but it binds to very big proteins that are circulating in the blood. So that creates like a macromolecules, and we do like that because one limitation with the blood-brain barrier is that the big drug that MDs want to use in clinic to treat the patient cannot get through because they are too big, and we do think that when we open the blood-brain barrier, depending on the size of the drug that you wanna inject, this barrier will not be open for the same amount of time. Meaning, if the drug that you want to inject is very small, you can end up thinking that the blood-brain barrier is open for a much longer time. However, it closed down, opens and closed down it's progressive, like a progressive closing of this barrier. So you need to find the right window for your drug, and depending on how big is your drug, you don't have the same organic window. So if you start to use a biomarker to somewhat measure how fast the barrier closes, and if you use a marker that's quite big, it's much better because at least it overestimate the time of this barrier. So that's why we decided to go with that marker. Here's some imaging we were at first trying to know how long it stays in the tissue and does it work. So here this control mice did receive the green marker but was not treated with the ultrasound. And this mice here received the green marker and was treated with the ultrasound. And you can see that when you use this ultrasound to open this blood-brain barrier, the green marker is getting much faster, much more. And then we did some, over time monitoring of this blood-brain barrier opening and this brain signal in our mice, just to try to understand how long it takes for the mice to fully eliminate this marker. So that was, preliminary data to try to understand how we can use this endocyanin brain marker to monitor the results. And I will finish with these slides. The next step, okay, we can use the brain marker to monitor the blood-brain barrier opening and closing, but now how fast does it close in our mouse model? So to do so, we did inject the mice with the same brain marker, and we treated them with ultrasound. The only difference here is that we injected the brain marker at different times. So here there is no delay. We treat the mice with ultrasound, and right after we inject the brain marker. Or we compare that with what would happen if we inject the brain marker 15 minutes later, or 13 minutes later, or 4 hours, or 24 hours, and how much of this brain marker, which could be a drug at the end, even if we are not there yet, how much of this brain marker is getting into the brain. And it seems that, it's like changing, opening and closing gradually. And the peak, where the signal is the highest, seems to be when you inject the brain marker, and again, that can be a drug, 30 minutes after you open the blood-brain barrier, 30 minutes after you treat the mice with this ultrasound. So we need to repeat this. That was done with mice, healthy mice that didn't, harbor any tumor, but that is laboratory developed a lot of pediatric brain tumor mouse models. So we will definitely leverage this resource in the lab and do the same thing with mice harboring pediatric brain tumors. But from those preliminary data, at least we know that the brain marker can be something we use. And then it seems that the window after opening the blood-brain barrier, after this ultrasound treatment, is quite short to collect our circulating biomarker or to treat the mice. So in conclusion, we so far have developed this ddPCR assay that Denisa showed you data targeting the most common viral pediatric brain tumors. The next step, and she started to show you some data, is to optimize this assay to try to lower the limit of detection, to detect lower and lower amounts of circulating tumor DNA into a patient. The second bullet point here is that we did show that it's feasible to find and detect circulating tumor cells in cerebrospinal fluid sample. Now the next step before fully calling them circulating tumor cells is to perform some genomic analysis and try to see what they can reveal. And the last step is the mouse work that I just showed you. And we did validate that we can use this brain marker to quantify for how long this blood-brain barrier is open, like what's our time frame to treat the mice. And yeah, we will now if that blood-brain barrier opening helps us to release more circulating biomarker in blood and CSF in this mouse model of the And with that, I will thank you for inviting us, all our sponsors. This is David's team, Expanding and Expanding. And of course, thank you to the Nina Foundation for giving us the chance to be here with you today.
We're done with the science, don't worry. I hope we're not too scared with the mouse work. And what we try to do, and showing that we are a team working all together, to show you that from the beginning we look at the disease, we try to understand the disease by looking at it first, and it gives researchers to have ideas to understand why is it different, the invasion thing, and how we can target it. And at the end, David mentioned that some of the drugs that I used you mentioned the MEK inhibitors in clinic can be targeting these invasion things, and invasion things is known from 1945. But now we have more biology, we can go from exactly these ideas of the invasion, understanding the invasion, to a drug. We try all the time, and that was Denisou's work, and thanks to the Foundation funding, she could come to France to help us with that, because we need people for that, to try to understand, to monitor this disease, and to be able to, not to do biopsy every 3 months, but to do this liquid biopsy work, because then we can monitor the disease and understand how the disease evolves. David showed you how the disease can evolve when you give radiotherapy and become radio-resistant. And all what Anne-Florence and Denisou showed you is maybe we can understand it in patients without giving aggressive procedures, just with liquid biopsy, just with maybe longer function, but even blood tests, because the ultrasound can open it and give what we call the sonobiopsy, to give access to what is in the brain, and to understand the biology at the same time, and then to open it to give a drug. So we try a lot of different things, a lot of different little steps. We didn't put everything, obviously, but step by step, you can start from an idea, invasion, to a treatment, to an idea, opening the blood-brain barrier, to monitoring the treatment, to monitoring the resistance. And all this together, we always need people, and we discussed this with Martin a lot, that if people are excited about that, want to spend some time in France and work with us, we definitely accept candidates' application for that kind of stuff. And also, not even candidates to come to our lab, but collaboration here. If people want to work with us, and we're open to all those things. And thank you a lot to Denisou Foundation to accepting us here today, but also to giving us the opportunity to work with Denisou, who is an amazing person. And I understand that it's really hard to have, I don't understand what you are saying, so I'm okay with that. But if it was in French, it would have been awful, so thank you very much.
Samuel, David, Anne-Florence, and Denisou, thank you a lot for your lectures. I hope that it was interesting for you. We are extremely happy that you have accepted invitation to these lectures and that you support both Foundation Nina and all the children who are affected with this disease in our country. I'm happy that scientists and you all came to us. Before I, let's say, open the speeches or lectures for discussion, we'll be very happy if you accept the invitation also for some small coffee break after the lectures. And I'm very thankful to National Technical Museum that has given us opportunity to organize the meeting here. And that they are always nice and open for cooperation. I'm also very thankful to Ms. Šternova and Ms. Fantastic translation, because it was quite busy work with not enough time for that. Thank you as well. It was a nice job. And so let's say the lectures are open for discussion. So anyone can raise the hand and ask loudly what's the question.
Well, I have a question. Can I? I would like to ask you, how long have you been working on your study? And I would like to thank you that for your coming. I really appreciate it. It was really awesome. So, and another question, do we know anybody who has been cured, ever cured, like completely? Or?
So for the how long date?
2013.
2013, so the program started, so the first biopsy started in 2007. So it's recent work actually, when you take into account that the disease is known for a while. And so, yeah, 2013 when you arrived and you started all this program with Jacques, it was with the first biopsy, it was the first clinical stuff. The last question is really hard because The question is, from this specific disease, to be completely transparent, we don't know exactly. We have patients who haven't relapsed for a while. The problem is, what do you call it cured when you don't have long-term results from that? So this is where we lack of data. So we have patients who have long-term survival of the disease. Very few patients, I would say, less than 3%, to be precise, actually. But we don't know if it's cured or just 2 years, 3 years, 5 years, and I think that's the most that we can say today.
Okay, thank
But
you.
we don't give up the hope that we will have.
Okay. And do you collaborate with the other countries? Because I've heard that, for example, in Spain they've started, similar study as yours. So do you share your results? Because more patients you have, more results you have. So then it's good to.
Yeah, that's a very good question, actually. It's a good question, and yes, so the BioMedi study. So we had the BioMedi 1 study, which randomized 3 different drugs with radiotherapy upfront in DIPGs and DMGs, and then the BioMedi 2 study. So the number 2, it's a BioMedi 2, that randomized everolimus with ONC201 with radiotherapy for every patient. And this study is open in France, but it's open in Spain, actually. And it's opening in different countries. It's opening in Belgium, Italy, I'm not sure, Sweden, for sure. It was supposed to open in the UK, but European Union being smaller than before, it's harder. And I know that Michal Zapotowski, who is a neuro-oncologist working here, is trying to open it here. So yes, you're right, the more patients, the better. And I will try to work broadly in Europe. However, we also collaborate, and I think David can talk about that, with US, not in clinical studies because it's harder, but maybe you can say some word about your collaboration with the people from Harvard.
Yeah, so definitely there are collaboration. It's not the right slide to talk about collaboration, but we do share samples because some people can analyze them a bit faster than we can as of now, and we have trying to find synergies, and to speed up discoveries. So we have collaboration in France, obviously. We have collaboration in Europe with Johannes Goyo, with in Vienna, with Marina Filbin in Dana-Farber. We are in contact with Karl Koschmann on some aspect as well. And sometimes we do discoveries side by side, that we share in these conferences. So it's definitely a crowd, a worldwide crowd working on these diseases. It's not so many groups in the world, and I guess we know most of them and interact with them quite regularly.
And last question, sorry for that. And how many patients are being treated right now in France? In that study you mentioned.
What do you mean being treated? We have 100 patients per year that are diagnosed now with DMG. We include pontine, thalamic, and spine, and we include adults as well, because the more we look for them, the more we find them, especially in young adults and adults. I think it's around 60% of pontine tumors, 40% of thalamic tumors, and thalamics are more in more aged patients. So within Biomed, I think inclusion are around 300 patients for Biomed 2 right now, but opening of the European centers is quite recent. So, and I think the age is around 450 or 500.
Yeah, something like that. One other thing is, we use, the BioMedi study that Jacques built with the whole team, with the whole, actually European Union, but not the whole, but a lot of different people, is one study, but there are other studies where we gather, or not even studies, like, for example, since we have the BioMedi study to get access to ONC201, that is a promising drug, even if it's not like a magical drug, it's still something that we need to explore. We also have a compassionate use program in France, and we register all the patients within a national registry that is in Gustave Roussy to get all the information from all the patients from France who got ONC 201. But in addition to that, some patients come from other countries in Europe to get the treatment because it's not accessible in all the European countries. In some it is, in Germany it is, but not in all the European countries. And we also register those. So we try to gather as much as we can, for sure.
Thank you.
Any other question?
Try to be as scientific as possible, but just maybe I missed it. What is the trial? What drug you use in the trial now? Are you using any drug or is radiotherapy?
Maybe I missed it, we didn't talk about it. So BioMed trial is a trial for patient upfront, so new diagnosis, where we propose to the patient, if they accept, to do a biopsy of the disease to confirm the diagnosis. I told you not all the patients are DIPGs or DMGs, so it's about 8% of the patients who has another diagnosis, and within these 8% of the patients, most of them are other kind of high-grade glioma, but different, and a few percentage of this is low-grade glioma, so completely different, not deadly disease. So we remove them, and then after this biopsy, the patient and family consent to participate to the trial that is upfront radiotherapy, that is standard radiotherapy that you can find everywhere in the world, plus after radiotherapy or during radiotherapy, it depends, randomization between everolimus, that is the winner of the BioMed 1 study with the best results, which is an mTOR inhibitor, against ONC 201. So the patient will have either everolimus or ONC 201. And it's not double blind, everybody knows, but it's randomization. Randomization, for those who don't know, it's a way to distribute the patient by a computer, not me, not the patients, just to be sure patients are balanced in both groups to be able to compare which one is the best. And then, if the treatment doesn't work, the patient have access to the other drug. So everyone at the end will have access to both of the drugs, but not in the same order. The idea to do that is to see if there is a difference between how long it takes for the disease to come back between everolimus or ONC201 plus radiotherapy, because radiotherapy works. Definitely it works. It just doesn't work long enough.
So anyone else before I try to move you for the coffee? I would like to ask you, so do you expect that the liquid biopsy will change the current strategy and that we'll stop to do the stereotactic biopsy and open biopsy to confirm these tumors?
No,
I'm not for it. It's yours.
it's yours.
I think it's somewhat the idea. I don't know. I think it's still gonna take some time before we get to a point where we can get as much information from liquid biopsy that we currently have for the tumor biopsy. But that's for sure the future, because you can imagine that having a blood draw from a child, it's much easier to do that in intracranial, like surgery. So that means also that you can do this blood draw weekly if you want to. So you can have like somewhat a real-time monitoring of the disease. So if we can manage to show that in this DNA and these cells or other synthetic biomarker, whatever we can choose, if we can get the same level of information that clinicians already have now from the biopsy, then they can potentially react faster and anticipate the next step and time is say that.
One more question, and this is the last one, because I really don't know if the coffee is prepared. I hope so. But I have too many questions. So I'm very happy that there is a centralized course that dedicates to these children with very aggressive disease, which didn't have the hope. I hope that someone has persuaded us that because we have very low experience, we didn't know a lot of things about the disease less than 2 decades ago, and now we understand more and more. There is a hope for the future, and that you really concentrate on that. So, do you expect that the ultrasound opening the blood-brain barrier can be used also for, say, monitoring of treatment, and also improve access for the drugs to the tumor cells itself? Are planning some clinical trial?
Well, there is some can answer better than me, but there is already clinical trials. This technology is not new, like using ultrasound to open the blood-brain barrier. It is done with adults. It is currently done in Paris in a hospital in pediatric patients, pediatric brain tumor patients. And they are, other subtype, not the one that we have been talking about today, but anyways, there is the idea that has been validated in adults, and there is a few preclinical studies showing that if you combine this ultrasound, that just opening the blood-brain barrier temporarily, it's non-invasive, you can, like the drug can get in easier, and then you can imagine that if the drug go in easier than, better than it is today without the ultrasound, then you can decrease the dose. If you increase the dose, you can potentially decrease the side effect and improve the efficacy. It's opening this blood-brain barrier and showing that it will work in pediatric brain tumors. If the barrier is open, the drug can get in, the circulating biomarker can get out, and then we can multiplex the results.
Já bych ještě měl jeden takový dotaz na Denisu, jestli by mohla říct, jak vidí svůj budoucnost, nebo jak naváže na tu zkušenost z Paříže dál vlastně krátce.
To je dobrá otázka. Je vlastně, já bych chtěla mluvit tady. A je pro mě momentálně těžké na to odpovědět. Je pravda, že si hledám práci teprve, ale ráda bych zůstala vlastně ve spolupráci s kolegy a bylo by to fajn. Ale mám vlastně teďka další 6 měsíců, co strávím ještě v Paříži, tak uvidím, jak se bude potom.
Něco doporučila by si někomu dalšímu, aby se zapojil do práce?
Určitě, je to super zkušenost.
Dámy a pánové, moc děkuji, že jste přijali naše pozvání. Já doufám, že se vám přednášky líbily, že vám rozšířili nějaké informace o problematice DIPG, DMG, o tom, co se dá v současnosti dělat, na čem kolegové pracují. Pokud máte nějaké osobní dotazy, určitě rádi přivítají a pojďte si s námi dát kávu, něco sladkého, slaného. Děkujeme.
Pardon.